Hello, my name's Christopher Bravery. I'm a consulting regulatory scientist at Advanced Biologicals. And in this talk, I want to discuss the common gaps I see in stability studies, generally for advanced therapies, with a little bit of a focus on viral vectors. As opposed to just discussing stability generally, I want to really focus on where the big gaps are right now from what I see. So the first gap is I think perhaps, possibly, I'm not sure, because ICH Q5C shouldn't be read in isolation. What you should really have done is read the ICH Q1 series as well. And going back and looking at Q5C, I sort of feel that I could see how you could miss out this first key issue if you had thought that because Q1A is written for small molecules, it's not relevant to read. And indeed, in the preamble for Q5C, it does say that the guidance Q1A applies in general. So what's the gap? The gap is demonstrating that the analytical methods that you are using for your stability specification are actually stability indicating. And this gap is really quite significant in that it feels like it's almost every stability section I look at has this gap in it for advanced therapies. Whereas for my biotech clients, it's not so common, but it does occur also. So it feels like this is a little gap that we really need to focus on a little bit because it's actually quite important and it will lead to questions at approval, which you might not have got during clinical development. So Q1A says, it talks about stress testing, and this isn't really mentioned very much in Q5C, so I could see how it could be overlooked. And the idea is it helps to identify the degradation products and to validate the stability indicating power of your analytical methods. And you only really need to do this perhaps for one batch, although that would be one batch at the beginning, and potentially you might want to repeat this for various reasons later in development. For instance, it might be a way of supporting comparability studies, although normally you would do accelerated for that rather than stress testing. So I'm not going to go through this slide, but there is a bit in there about stability indicating profile, but he doesn't really talk about the need for the stress testing in quite the same way as Q1A does. So rather than pick out various bits and bobs from the guidelines, here's my view about what I think you should do, what is useful to include in your dossier, and what is useful for you as a developer to understand about your product. Remembering that what you're wanting to understand is both in the way in which your product degrades, but also the rate at which it degrades. Remembering that certain quality attributes might change more quickly or more slowly. So some may be more or less stability indicating, or the period at which they start to show a change might be quite different. You need to find some conditions that allow for a good rate of degradation that's convenient for you to characterize. So sometimes I see studies done that take hours, and in some cases that is necessary. For instance, if you've got a genetically modified cell that is frozen, or even if it's not frozen, the in-use stability is going to be hours at room temperature, and therefore you've got rather little choice. But when you're doing a forced degradation study or a stress study, then it's up to you really to try and find the right conditions to make it convenient to do the characterization. So my feeling is probably you want to look at something that happens in days, over a period of days rather than weeks or hours. But that's completely up to you. You can do it over a few weeks if that's preferable to the way you're working. Of course, how you come up with the right conditions to do that is what's the right temperature, for example, because temperature is the most common way to do this. That's either based on prior knowledge or, you just have to have a go and see what happens. The other issue, I think, is that when people do accelerated or forced degradation or stress studies, these sort of or in use, they often choose too few time points. So you're losing a lot of knowledge and data by not following it over enough time points to see what the trend is, remembering that it's fairly unlikely that the product's going to degrade in a linear fashion. It's almost certainly going to have a more complex shape than that. And the crux, the real missing bit is actually having sufficient time points that go completely out of specification. So there's no doubt that now the product's fully degraded. And quite often I see so-called stress studies that actually don't reach a point where any of the parameters go out of specification. So it's not really very convincing. So I think you need at least 4 time points, and I would recommend that you try and have at least 2 time points going well out of specification. I mean, 50% degraded, it would be great, but 25 might be enough, but it's got to be well outside your specs, and there's absolutely no doubt that you've degraded it. And then don't just use the quality attributes that you've chosen for your randomly for your release specification based on what you're using. Sorry, for your stability specification based on what you're using for release, which is to me seems the way that many do it. They just, they've got a sort of a slightly guessed release specification early in development. And then from that, they just guess which ones might be stability indicating, knowing that certain things should be there. Do some proper characterization, because you will learn a lot more, and you might identify attributes that you hadn't thought of that might actually be useful also for release. So it should really be full characterization and not just a few tests. The other big gap might sound like a funny thing to say in a stability talk is formulation development. I say this because, one, I see rather few formulation development studies for advanced therapies. Some are more likely for gene therapies than for cell therapies. Cell therapy developers almost never seem to do any sort of formulation development studies. And there are various reasons why this is actually not useful, or is sort of detrimental to your overall development. For the purposes of this talk, you would learn a lot about the stability of your product from formulation development studies. Of course, that assumes that you've done some forced degradation, you got an idea what to look at, which parameters to look at. But those sort of formulation development studies where you look at different storage temperatures, pH, osmolarity, all that sort of stuff, teach you a lot about the stability of your product. And ultimately, while you may not mind going into a first-in-human study with a suboptimal formulation, that's not uncommonly done. But I think, the COVID vaccines, I think, have rather emphasized that if you haven't got the time or don't spend the time to do formulation studies, and in this case, they probably didn't have the time, they've gone onto the market, if you like, with Pfizer-BioNTech, for example, is on the market with what was probably only ever intended to be a first-in-human formulation, because they didn't have time to do more. Although I might be wrong, it might not be possible to store that product at a higher temperature. But clearly, sub-zero temperatures are not very convenient. Now, in the case of the COVID vaccine, of course, we were trying to immunize millions and millions of people. But you might be working on a very rare disease, and you might say, well, it doesn't matter that, this is only going to a specialist center. They will have the freezers, they can deal with it. But it would still be a lot more convenient to them if the temperature was a more convenient temperature for storage. And the overall dosage form was designed in a way that's convenient. And if you try and rush through development, these are the sorts of things that fall through the gaps, and you end up with a suboptimal formulation. So, for example, actually needing to dilute or reconstitute or whatever is a sort of a suboptimal outcome, because there's a risk of breaching sterility and so on when you do that. Of course, the optimum outcome would be a pre-filled syringe, which is probably not going to happen just now. But it might be possible eventually for viral vectors. I'm not sure. I've not seen anybody try it. But all that, you learn a lot from doing those formulation development studies. And so just copying the formulation of others or sticking to something very innocuous like saline is just not going to be ideal for the commercial phase, and you will miss out on a lot of knowledge. So what sort of tests should be there? And I'm just going to whip through this list simply because I do find sometimes the list of quality attributes is a bit odd for stability studies, and often some of these things are missing. Actually, I'm going to start at the bottom because extractable volume seems to be quite commonly forgotten. It is a requirement actually for parenteral products to have extractable volume. Clearly, it's much more important if you've got a pre-filled syringe where you administer the whole volume, but it's still expected that the volume you state on the container is the volume that can be extracted from the container. And don't make that mistake. I had a client that made that mistake very recently, where they were quoting the volume of the container being what they put in, not what could be extracted from it. Funny little mistake that maybe I've overlooked on other occasions, but it should be your extractable volume. Now, the idea of testing that throughout stability is clearly that you want to be sure that you can still extract enough of the stuff to deliver a dose to the patient. If your volume is going down, it will be an indicator that you've got probably a breach of the container closure, and you're losing water, which can occur in a freezer as well, of course. Going back to the top, purity or quantity should always be there, because again, you want to be sure that the amount of that there's enough product in there that you're administering the intended dose when you withdraw whatever the volume it is on the label for the patient. Potency, we definitely want that to be, we definitely want the product to still be potent at the end of shelf life. But the potency assay may or may not be stability indicating. I mean, you would expect it to be stability indicating up to a point, but it just might not be very sensitive to change. And I don't think it would be unreasonable if you got a potency matrix. This wouldn't be the case if you only got a single potency assay. But if you've got a potency matrix and one of them isn't stability indicating, then there's not particularly necessarily any need to do it, or maybe you would just do it at the end, like you would do with sterility. Sterility and endotoxin, obviously important. It is possible to omit sterility testing in a stability protocol, sterility, oh gosh, as long as you have a sort of routine container closure integrity testing, which is probably not which is not so easy to do with low temperature frozen formulations. So probably easier if you've got a fresh formulation. But it is in principle possible to do it that way. Other tests should generally focus on the product-related impurities. Process-related impurities, well, you're not expecting those to change unless there's something weird in there. And then the other one that people don't always think about, although for the most part there isn't really anything there that's too worrying, that necessarily needs to be looked at. But I know with viral vectors and with viral vector gene therapies, sometimes excipients are being used to sort of promote the transduction of the intended target tissue. And those excipients, if they're somewhat more complex, so the most common complex one might be human serum albumin, they will have their own stability, and you can't have a product stability of longer than the excipient stability. So you do also need to think about the stability of the excipients where they're complex excipients that might degrade. What other issues do I see? Early in development, one of the issues that I think comes up pretty often is somehow overlooking the fact that you might have stability data from the non-GMP process from a research material. And indeed, you should have, really, if you haven't been looking at the stability of your product during the preclinical phase, then you're missing a huge opportunity, and you're actually diminishing the evidence you have for whatever the stability you claim is at for first in human. So there's nothing wrong with including those data. You're not saying that the stability is going to be exactly the same, because the container will be different, the excipients might be different, and indeed the process was probably different. Certainly not the GMP process, perhaps, but it still provides supportive stability data, so that the paucity of data you tend to have for first in human, which could be in some cases as little as one batch, although in other cases maybe a couple of engineering runs, but usually only a few months stability data is available. And sometimes it's actually the limiting factor to submitting the initial first in human dossier. So do use that data to support your stability conclusions, and maybe use it to support a modest, very conservative extrapolation of your stability data. I.e., if you got 3 months, then you could say we consider the shelf life 6 months at this point in time. We'll let you know if anything goes out of spec. But remember that in general, we don't accept extrapolation of stability data for biological products, in part because they don't tend to follow a linear degradation. Then for various other sorts of stability studies, I think one of the most common problems I see in all manner of other sort of stability studies is just too few time points to actually be very useful. A stability study with 2 or 3 time points is just not particularly helpful. 2 points is always a straight line. 3 points is pretty easy to fit to a straight line. You need really at least 6 time points to be able to see the shape of a degradation profile. And with your real time, you really need quite a lot of batches eventually to be able to look at these patterns and see whether they're consistent, because, you know, we all know that one particular set of stability data might give you one impression that, I don't know, that you've got a sort of linear downward curve that, you know, is quite rapid or something. And then another time it looks like it goes up a bit, and then another time it does something else. But if you look over multiple batches, eventually you might actually see a specific trend, and you might well notice that isn't linear. And it also builds sort of a pattern of understanding. And remembering that, of course, individual time points sometimes go a bit wobbly for various reasons, although obviously if there's a significant wobble, you need to look at it. But those patterns, you're looking for those in part to make sure that they make sense, make sure that there isn't something you're overlooking because that sort of weird degradation. Well, why is that happening? You know, you should be exploring it and trying to understand why. As I've already mentioned, no time points going out of specification. Try and, you know, for stress, for everything other than real time, but even real time, it might be useful. It's nice to see the stability study go to the point where it goes out of specs, and that you know that you know everything's on that your stability specification is working, if you like, but it also allows you to see the ultimate trend. I mean, that might not be possible for everything, but not having it in any studies, which I see quite often, goes back to the first point of this talk, really. And then lack of consideration for other types of stability study and the value that they might bring. Photostability, you might be surprised. I used to think this seemed daft for biotech products, but actually it does matter. Photostability for your excipients might matter as well. Agitation, that sort of thing. And that's it, really. That's my walk through my my top issues with stability studies, and thank you for listening.